Okay, I take this honor to welcome you all. My name is Nabende Adam, and I've always been your facilitator for biology. And of course, this session is hosted and sponsored by Holistic Learning Platform. All these lessons can be found on YouTube, Holistic Learning Platform. You can follow all of them from Senior 1 to Form 6. You can always go there, watch them, put some comments, comment even about the videos. It is better for us to make a better platform for learning. So you can always do that. For tonight, we are going to go through growth and development. I'm sure it's what we covered the other time we met. We were unable to meet last week, given the nature of the country, because we had some elections. Of course, where we underwent internet shutdown and we were unable to meet. But hopefully we are going to meet and then we cover what we were supposed to cover last week. So tonight we are going to go through growth and development. This is a senior 33rd term topic, which I assume some of you have done or have not. So we are going to go through it together as we understand how this comes up. Last week we covered partly what happens during growth and development. So today we shall try to go into deep detail. And before I start, I will request someone to put up the hand and give us an opening prayer. Namulondo. Okay, there are very many here. I don't know. I think Namulondo talked. We should pick another person. Okay, pray for us.
Let's pray. Our dear Father, we bow before you because you are holy, you are all in all. At this time, Lord, you are going to study. Be with us, send your Holy Spirit to guide us. I have prayed through your Son, Jesus Christ. Amen.
All right. Thank you for that wonderful prayer. So you can put down the hand now. Okay, put down the hand, Florence. Good. So, if you might remind me, because it has been some time now, I hope we were covering this topic, growth and development, before I can start. Was this the topic we were covering? It must be the one. So, the other time we handled partly of how growth comes up. We explained these terms: cell division, cell expansion, and then cell differentiation. So we shall today try to go deep into development. We handled this partly, last week, the growth part. Also, as a point of information, we shall not abuse the chat room. Okay? Meaning, I don't want to see the chat room loaded with a lot of messages not discussing biology or what we are covering today. Should I say that happening, then I'll ask the technical team to turn it off such that we can concentrate on the lesson. So on that note, I don't want to see a lot of questions in the chat room. Should you have something to share, then you put up the hand, I'll let you talk. Like I always said, this lesson is yours, not mine. So you have to be at the center of the learning. I'll not read off any messages from the chat room, but instead, I will be requesting you to put up the hand, okay? So don't abuse the chat room. Okay, so I'm just reminding you about what we covered last week. We talked about growth. We said that growth is the increase in dry weight. We said dry weight means the weight of something when all its water has been removed. We talked about terms like cell division, which simply means cells are dividing. One cell is dividing to give two cells with the same chromosome number. if it is occurring by mitosis. Okay, someone has put up their hand here. Oh, okay. All right. So put up your hand when you have something to say. Otherwise, don't just put up the hand for fun. So you have cell expansion. Here, the cell increases in size because of uptake of water. We know that when a cell takes in water, most a plant cell, it gets turgid, meaning the cell vacuole presses against the cell wall, and therefore making it elongate. And when it does that, shall say it has undergone cell expansion. And then lastly, cell differentiation, which means cell specialization. In this case, cells become specialized to perform specific functions. So that one is also important for efficiency of an organism. So growth is usually accompanied by an increase in the complexity of an organism, which is also called development. So development is the change in form and complexity of an organism. In other words, development comes because of cell differentiation. So we did this last week, the differences between growth and development. I'm also assuming we are not here to take notes. I'll endeavor to explain. Where you don't understand, I'll repeat for you using the best example you can. I can for you to understand. Then I will share the notes later. So your role is to pay attention. Where you don't understand, you ask. And we always say that this is a fully diverse class where each one has a right to ask as long as it is related to the topic. Okay? So don't fear that because you're from this part or you're worried that someone will laugh at your English or accent. It's not that. Here we share freely. Okay? So mitosis and growth. So you have a process called mitosis. In mitosis, one cell divides, the mother cell divides to give us two daughter cells, and each with the same chromosome number as the parent cell or the daughter cell. This one means that like all cells have the chromosomes which are found in the nucleus, and chromosomes are the ones carrying the genetic information. So in mitosis, one parent cell shall divide to give us two daughter cells with the with the same chromosome number. Meaning, if the parent was having, let's say, 23 pairs, then the daughter cell shall also have 23 pairs. So in other words, during mitosis, the characteristics are kept. Meaning, for example, if we would take mitosis taking place in the cassava stem cuttings, then you'll get exactly the same cassava, which the cuttings were got from. Why? Because the same chromosome number is maintained. This is very different from the one which occurs in human beings. For example, if human beings are also reproducing by mitosis, the first thing is that we would only need one parent. Okay. In other words, a mother would only be needed, or a father would also be needed. Then the father would give birth to sons, and then the mother would only give birth to daughters if it was by mitosis. Okay. Someone has something to say? Please, anytime you put up the hand, you be quick. Okay.
Excuse me,
Yes.
teacher. You're breaking.
Oh, my network.
Yeah.
Okay. Yeah, I think that's my network, but I hope it becomes stable, of course.
So could you please repeat that statement of the parent cell and what it produces?
You said that during mitosis, a parent cell divides to give us two daughter cells, and the two daughter cells have the same chromosome number like that of the parent. Okay. I gave you an example that, for example, in stem cuttings of cassava, if you get stem cuttings from a particular cassava plant, then the young ones shall be exactly like the parent, meaning they have exactly the same chromosome number. But say this one doesn't take place in human beings, for example, during reproduction. Because if it would happen, it would mean that one, we would need only one parent. So meaning we would need only a dad or a mom. And then a mom would produce all girls, and then a dad would also only produce boys. But say this one doesn't happen. in human beings. However, it happens in human beings in a time when cells are increasing in size. For example, should you have a particular part of your body cut, or you've got a bruise, like you've been involved in an accident, then the ability for the cells to grow back, they will grow back by mitosis, where one cell shall divide to give us two daughter cells to fill up the space which has been created by the accident. Silence?
Excuse me, sir.
Yes, please.
So why is it that when they divide, they still have the same number as the parent cell? And why is it only divides into two?
I note how many. Okay. Now, mainly cells divide by two processes. One, mitosis. The other is meiosis. Okay. And if it is by mitosis, okay, one cell, the parent cell, shall divide to give us two daughter cells. And the two daughter cells shall have the same chromosome number like the parents, eh? Otherwise, because it occurs through one stage. But for meiosis, it gives us four cells, meaning they first divide into two, then the very two daughter cells divide further to give us other two daughter cells, therefore ending up with four in one process. So in mitosis, one cell divides to give us two daughter cells with the same chromosome number. Okay?
Okay, thank you, sir.
Blessing? Be quick when you put up the hand.
I was just asking,
Yes,
like when
please.
the parent cell produces the daughter cells, do those
Come again?
daughter like when the parent cell produces the daughter cells, do those
Yes.
daughter cells like also become parent cells and they also divide into other daughter cells or something?
Exactly, yes, because life is continuous. Okay? Much as there is aging in cells, that's why you see you can wake up in the morning, you bathe, then by evening you see some, when you scratch your skin, you see there are some cells, meaning they're also dead cells. Cells also have a lifespan, eh? So that is also important to note. Josh?
If the mother cell gives birth, does it destroy up?
Come again? Come again?
If the mother cell gives birth, does it destroy up? Destroy up.
Now, for the first thing is, it don't say it gives birth, but rather it divides. Okay, the critical word is it divides. Okay, of course, you say that when that cell becomes old, because all cells have a lifespan for which they can live. So when that time for them to, I mean, when that lifespan of their cells, I mean, of their time, or when the lifespan comes to the real time, then that particular cell must die.
Okay.
Okay.
Thank you, sir.
Xiaomi?
Excuse me, teacher.
Yes.
I wanted to ask whether this process only happens in all plants.
It happens in all living things.
Okay. Thank you.
Jovan?
Good afternoon, teacher.
Good afternoon to you.
Teacher, I would like to ask this process of mitosis.
Yes.
Teacher, does it always take place in a living organism as long as it's growing?
Exactly. Because the main purpose of mitosis is growth, okay?
Yes.
So as long as a particular living thing is, let's say multicellular living organisms are still alive, then they must continuously divide. Just that, of course, as you age, for example, for you, you're still adolescent. So usually you're growing, meaning the number of cells which are being made, or the new daughter cells which are being made, are far more than the ones which are dying. Okay, that's why you
Okay.
grow. Then for an older
Now,
person, the number of living cells the number of cells of daughter cells which are being made might not be the equal to the ones which are dying off. That's why you see that their skin starts shrinking and they get wrinkles.
oh,
Mm.
now, teacher, what I
Yes.
am wondering, our bodies are made up of
Mm.
millions and millions of cells. Teacher,
Yeah,
now
yeah.
if every cell is dividing to two daughter cells,
Yes.
don't they reach an extent and they are excess in the body, or how is it regulated?
Exactly. If this is not controlled, that's when you hear that someone has got a cancer or a tumor. So all cells are regulated at a particular point, which is done in the nucleus. Okay, that's why you can never have the right hand being bigger than the left hand. In case that one happens, it means that there is some miscommunication in the cell processes, which is commonly classified as a cancer. So all these cell divisions are controlled.
Okay, teacher.
Mm.
Teacher, now how are they controlled, or what system is responsible for that?
One, we have the cells dividing being controlled by the nucleus of a cell, but also this is coded in the DNA. That's why you can never have, let's say, a human being who becomes so tall, like they become 100 meters tall. No, it is also coded in the DNA, such that they know that this particular person must have this number of cells being made. Okay. And also there comes a time when someone cannot grow any taller, for example, meaning in their codes, their growth ends at that time. Okay. So, but secondly, also there is a
Yes.
system which is removing the old cells, the cell debris, meaning for any cell, and depending on the number of cells dying, it shall also determine the ones which are being made. For example, let's take an example of red blood cells or another cell. Should it should it be nearing its lifespan, then it will
Yes.
develop a signal on its cell surface, which shall signify the immune system. I mean, it shall let the immune system know that that cell is old, therefore it must be removed. So when that cell is being removed, new cells must be made to replace it. So
Yes.
the control of the cell division or the making of new cells is controlled by a number of processes in the body. But the cells dividing is controlled by the nucleus, which is having DNA that determines a particular number of cells which must be made. Okay?
Thanks very much.
All right. Okay, I see this is becoming so long. I'm only letting 3 people talk. That is, I don't know, this is a hard name for me. Moixy. Please unmute. Are you there? Unmute?
Excuse me, teacher.
Yes.
I was just asking, since snails, like, they are, okay, like snakes can produce without mating with another. I was asking if they also go through mitosis, like to produce.
Snakes produce without mating?
Snails.
No, that's
No,
wrong.
I was meaning snails.
Snails, okay, maybe. But they all do that. Okay. So there are many, of course, there are many types of reproduction that we shall cover on maybe in second term holiday. So you're asking that do they also undergo mitosis? Now, to simply put it, mitosis is mainly for growth, okay? But usually for cells which undergo, like where we involve two parents, such cells must come out because of meiosis, okay? So anytime you're talking about growth or anything which grows, must undergo mitosis. Their cells must undergo mitosis, okay? Then
Yes, so
for cells which are going to lead to reproduction, for example, the sperm cells or the egg cell, they come up because of meiosis. and I'll explain why one time maybe. Okay?
Yes.
Sharif? Okay, let's put this to the end, otherwise I know we shall not finish. Yes, Sharif, be quick.
Hello, teacher.
Yes.
Teacher, is mitosis that's another name for cell division, or they are different things?
How can I say? Okay, cell division has two types. It has mitosis and meiosis. Okay.
So it's a type. Why do they have the same okay, the definition with cell division is the same as mitosis?
Of course, because it is okay. It is different. Cell division is increase in number of cells. Okay. And the increase in number of cells can come because of mitosis or meiosis. But the ones which are leading to growth come by mitosis. Okay.
Yes. Teacher, I thought even
Understand?
meiosis leads to growth, but just because this one has two processes.
Usually meiosis is for, of course, they all lead to growth, but meiosis is more used in reproduction, where we have the number of chromosomes.
Okay.
For
Yes,
example.
thank you.
All right.
Teacher, you can say.
Sharif?
I wanted to inquire that how are the chromosomes adapted to carry the DNA and genetic material?
Okay, so Sharif wants to know how are chromosomes adapted. Now, chromosomes are, of course, thread-like. Depending, they can be chromatins, but usually they are double-stranded. Something I don't want to go through so much. So they are joined by some bonds which link them together, and then they are carrying those genes within there. Maybe we shall discuss a lot of them way forward when we reach in the near future there. Otherwise, for now, shall just take it that they have those characteristics of carrying genes from the parents to the daughter, to the daughter cell. And so they just open up like a bridge, and then the other one runs along, copying the information. So for the mother, it shall only pick one side, then for the father, it shall pick one side, therefore joining them to form an offspring that is having characteristics of the mother and one for the father, depending on the one which is more dominant. Okay, I think let's focus on this for now, okay? I will answer on the questions in the next phase. Okay, so mitosis contributes to growth of organisms in two ways. One, increase in the number of cells in the body of an organism, and this is the basis of growth, and then also replaces damaged and dead cells in the body. So that one is also very important. And then let's go through stages involved in the process of mitosis. So here we shall pay attention. So the first stage is called the interphase. And then during the interphase stage, here the cell prepares to divide. During this stage, the cell organizes the food reserves. There is also replication of DNA. So all those ones take place, but this is usually a preparatory stage. It just prepares, just like when you're organizing for Christmas or any other event, or organizing for Ramadan, or organizing for any event of your religion or culture, you often go buy food, preparing for it. So interphase is also a preparatory stage. And then in nuclear division, the nucleus divides into two daughter nuclei, and this stage occurs in 4 phases. Here we have prophase, metaphase, anaphase, and telophase. So after interphase, 4 stages follow. And then you have the last stage here called cytokinesis. Cytokinesis here, the cell cytoplasm is then divided equally to form two daughter cells. So like we see there, here the cell divides to give us two daughter cells. So from this, from one cell, now here it divides to give us this, and that process is called cytokinesis. Okay. So let's go to importance of mitosis in living organisms. It increases the number of cells in multicellular organisms, which results into growth, and that is very important. So cells, or someone can grow only by mitosis when cells divide to give two daughter cells. Oh, someone put up the hand and they put it down. Okay. So it also replaces damaged or worn out cells. We said cells die, but also at times new cells which could still be young can be affected by maybe injury. Therefore, they must be replaced by mitosis. Yes? Joel, Chris?
Excuse me, sir.
Yes.
I was just pardoning, like you will clarify more on the stages of mitosis.
Okay.
I didn't understand them well.
Okay. Mitosis has 4 stages. I mean, majorly there are 4, but we have a preparatory stage called the interphase. Interphase is that stage when the cell prepares to undergo nuclear division. So during the interphase stage, there is organizing the food stores. There is also replication of DNA. In other words, the DNA duplicates or makes another copy of itself. And then after the interphase is sometimes called the preparatory stage. Then there comes the nuclear division, which occurs in the 4 stages. The first stage is called the prophase, and then the next one is called the metaphase, then anaphase, and lastly we have telophase. Telophase, we also have a key important thing which is called the cytokinesis. Cytokinesis refers to that time when the cell actually divides into two daughter cells. But meanwhile, up here in these other cells, there was simply nuclear division taking place. So lastly, after the nuclear division taking place, that's when it divides completely to give us the two daughter cells here.
Okay, I've understood. Thank you, sir.
All right. Chris?
Good afternoon, sir.
Good afternoon to you.
So good afternoon.
Good afternoon to you.
So I'm also asking on the stages.
Good
afternoon. Like I'm also asking about the stages. Like
Ask.
My internet had some problems when you're working on the stages, so I'm kindly asking you to repeat them for me. Hello?
Yes?
So I'm kindly asking you to repeat for me the stages. I didn't understand them well. My internet got some problems.
Okay, we say that we have mainly 4 stages of nuclear division, okay? But the first stage is called the interphase, which is the preparatory stage. It is preparatory in the way that it is not actually a cell dividing or nucleus dividing, but it just prepares for the nuclear division to take place. Meaning, here we shall have, during interphase, the cell organizes the energy stores. Then there is also replication, or there is duplication of chromosomes, or the chromosomes the DNA makes its copies. And then the other stages are called the nuclear division, and there are 4 of them. We have prophase, metaphase, anaphase, and telophase. And then lastly, we have a part called cytokinesis. This is when the cell completely divides to give us two daughter cells. Samsung? Please, if you talk, you lower down the hand so that I don't mix it up. Joel or Chris, lower down the hand. Yes. When you see me request you to unmute, you unmute very fast. So you don't waste a lot of time. Yes.
I have a question.
Yes.
Like sometimes we get wounds, and there's a point that said about healing of wounds, and
Yes.
through the process of mitosis. So,
Yes.
is there any way of increasing the process of mitosis so that the wound heals faster?
Of course. Meaning your reason. I mean your reasoning it out that is there a way of increasing mitosis such that the wound heals faster? Hope it's not like that. Otherwise, usually you can't, because all this is controlled by the nucleus, so you can't force rapid cell division. Why? Because much as it can happen, you will make mutations which will lead to cancers, which is not required. So usually what they do, you can minimize on the things which can deter cells from dividing. For example, keeping the wound dry so that you avoid fungal infections, such things. Otherwise, you cause the cells to divide very fast, then you can easily end up with a cancer. But should it be done controllably under biotechnology, it is also good, so you can quicken it. Like you have ever seen, there are nowadays Vaselines, which can, and lotions which can restore your skin back so that the scars disappear. So all those ones can be induced using bioactive substances, which can be put in smearing oil or Vaseline to help quicken the cell division. Yes, iPhone 17 Pro Max. Yes. Okay, that one was just advertising the phone. Malik?
Good afternoon, sir.
Good afternoon.
So I have a question.
Yes.
Sir, I'm asking, how long does mitosis last in the human body?
You can search that up and then you tell us using the internet.
So another question.
Yes.
Sir, can a human being like exist without mitosis?
They can't, because meaning, remember, cells are being made and they are dying. So when you're not making new cells, you die.
Okay.
Mm.
Thank you, sir.
Meanwhile, search that and then you tell us. Yes, MOIXY. I don't know what that. You'll tell me that name so that next time I don't find that issue. Yes? Okay, they are not there. Next one here. Malik, you can lower down the hand now. Jackie? Jackie? Faith?
Good afternoon, teacher.
Yes.
Okay. Yeah, my issue is that you rarely ask questions. Rather, you don't ask. Since the last lesson, you've not been asking questions. And then, okay, personally, I researched. And then the importance of mitosis, I had some of them. But then you teach and more teacher-centered.
I was teaching alone.
Yes, so I was not asking of questions, and some of us, researched. So when you don't ask questions, we lack the morale of researching again.
I rarely do that. My lesson is always completely learner-centered unless there are some interruptions, maybe. But meanwhile, what do you have to share with us?
No, like you already showed them up on the screen, so it does not make any sense. I thought maybe it would be like anybody who has an idea, then I show up. Yes.
Yes, that's why I want the idea now. Maybe because sometimes don't put up the hand.
No, because when you're teaching, you scroll and you go on the hypothesis already. So that shows that there is no
So
asking
do you want to show? So did you have any idea to share?
Yeah, I had ideas, but then I'm seeing all of them there.
No.
I had a...
Oh, you need to tell me wanted to tell me the importances of mitosis, right?
Yeah, I had to.
No, you can still tell us. Maybe you bring it in your own scenario. It is okay. So I'm sorry for doing that.
Okay.
Maybe next time better.
Okay, yeah.
Yes, praise.
Yeah, to ask.
Yes.
I like to elaborate more on the that can like elaborate more the things of inside of process and affairs kind of thing.
Oh. What I've understood is you want me to elaborate more on interface, metaphase, all those ones, eh?
Yeah.
Okay. Now, during the interface is the preparatory stage, where we said there is the organization of energy stores, there is replication of chromosomes, all that happens. Then during prophase, there is thickening of chromosomes. So during the interphase, we usually call them chromatins because the chromosomes are not actually seen. They are thread-like, okay? Now, during prophase, they thicken. I think let me get here a video such that we can easily see this. So during the interphase, during the metaphase, the prophase, they thicken up. Plan, and then you write the key points or the map of the content map of what is required of that question. Trust me, you won't miss out any of the marking points you've written. So that's the only difference you'll make from a person who has always been cramming the content but has not understood how to plan to bring out the content. So here. Okay, I want to get a short one for you. Okay, I don't know if you can see it there. So I'll explain for you how it is from the start. So at first, that is a cell which has not divided yet. So when the cell is starting to divide, you see these are called centrioles. They move to the opposite poles. These two moving. You see, they are moving to the opposite poles. And then this the things at the center here, which are purple, are the ones which we are calling chromosomes. Okay. So at prophase, they thicken. This one began from prophase. So here they thicken, but at first, during interphase, these ones are just thread-like. But during prophase, they thicken, like you see the way they are like that. And then during metaphase, they form one line. So you see, at that point, this is the point that we call a metaphase, when they align themselves along with the equator of the spindle. These ones are called spindle fibers. These ones running, going up. They are called spindle fibers. So at the point when these chromosomes align themselves at the equator of the spindle, that's when we call it metaphase. Then the next one is called the anaphase. At anaphase is when they start parting company to go to the opposite poles. That is called anaphase. Then the next one here, when they start dividing, we call it, that's when the telophase comes in. When this one goes this side completely, and then this one also goes this side completely. But it depends on the type of cell. So here, this one, in this way, it is taking place together with cytokinesis at one time. Now, that point when it is dividing completely, that is the one we call cytokinesis. So we have formed two daughter cells now. Okay. There are people who are putting up their hand, but they don't talk. Aria?
Good afternoon, sir.
Yes. Good afternoon.
Okay, I'm asking that in case they are setting, okay, like in which way can they set about the stages of
About this.
mitosis? Yeah, about the stages.
Of course, definitely. Maybe they can ask you someone was involved in an accident, okay? And then they are recovering. It is possible to be asked it is possible to present in your response how they can recover. Okay? They recover by the cells dividing by mitosis to give new cells, thus healing the wound. Maybe we can, I don't know, if we can come across such items, maybe we can do them. But otherwise, that's the way it can come up.
Okay, but okay, for the way you've explained the processes, the stages,
Yes.
it's kind of okay, it's brief, but in case they set the scenario, should we also answer it like the way you've explained the stages?
Now, you know that in the new curriculum, like, the things come in form of elements of construct, okay?
Yes.
Yes. So, meaning, in that particular element of construct, we cannot have cell division because this one now will be under processes, okay? That's why the most important thing is I'm putting it at the level of leading to growth, by not describing exactly what happens during that stage, though it is important for your knowledge also.
Okay.
So that's why I'm focusing more on the product of causing growth rather than discussing what actually takes place during cell division.
Okay. Thank you, sir.
All right. Yes, there is another person here. When you put up the hand, you unmute. After
Sir.
talking, you unmute. Yes.
So when I'm reading through the importances, this one that says it helps to generate body parts. Why is it when a human body part is cut off, it doesn't come back?
Okay. Now, there are complexes, just like if you cut off someone's hand, then it can't come back. Why? Because a human is more at a larger level, okay? More specialized and like when you learn about development, you shall learn that in humans or in most upper mammals, development occurs at a specific stage. Development means differentiation. So meaning even your body is differentiating. For example, there is a differentiation of the arm into the hand and the different fingers, which occurs at a particular stage of development. And should that stage be missed by mistake or whatsoever, maybe someone takes tablets and then some of the processes are disturbed, the baby, then the fetus fails to develop, it means that such people will grow without that part. So for humans, they are a bit of more developed at a different level. That's why here also in the regeneration of lost body parts, we clearly put starfish. For starfish, if you get a starfish, then you divide it into 1,000 parts, then you'll be having 1,000 starfish in the future. Unlike for us humans, you know that for us we are more developed and therefore growth doesn't, you giving birth or even other animals which reproduce using two parents cannot do that. That's why for us it is a bit different. iPhone Pro Max, please, anytime you put up the hand, you talk very fast.
Master,
Yes.
I would like you to enlighten me on the significance of meiosis.
Come again?
I would like you to enlighten me on the significance of meiosis.
We are covering mitosis, or you just want to know something about meiosis?
Yes.
Okay, meiosis is important. One leads to variation in the way that the offspring, the daughter cells being formed are different from the parents. Therefore, they become variant, so they be different from the parents. Secondly, it is important during reproduction. Why? In the case where the sperm
Baby is born.
cell is not halved, then we shall end up with a mutant baby at the end of the day. So that's why it is important for cells to undergo meiosis, either they are halved, and when the egg fuses with the sperm, then they can give a fertile offspring that is diploid. So the main importance of meiosis is variability or causing variation in offspring. Kiara, and you must be the last.
Okay. It's okay. Thank you, teacher. Teacher, I have a question, but I don't know if it's related to cells or what. I have come across baby being born with 6 fingers and others having like abnormal umbilical cord. It's too big, and I don't know if it's related to cells or other biological term. Could you enlighten me more? Thank you.
Now, some of the conditions are caused by, of course, they are genetic in the way that you can pick them from the parents, okay? While others can be got at birth due to maybe negligence or hygiene and others. But most of the conditions are picked from the parents. Like those, having many fingers or having the 60th finger, like that.
And even the umbilical cord being too big, like beyond the normal size.
That one, in genetics, I've not seen it. Therefore, it could do. I'm not sure if it is genetical, though I might attribute it to genetics. but it could be genetics, literally, because it is not a medical condition, it is just a disorder.
It's okay. Thank you, teacher.
Yeah. Okay, that one was the last. So you can lower down the hands and proceed. Otherwise, we shall not finish. So we talked about maintaining genetic stability because the daughter cell has the same chromosome number like the parent. That's why the chromosome number is maintained. I give an example that if mitosis was taking place in humans, then a mother would give birth to daughters, then a father would give birth to sons. But because for us we have meiosis take place, that's why a mother gives birth to sons. We have talked about regeneration of lost parts, and then we also have forms the basis of asexual reproduction. Asexual reproduction is the type of reproduction where only one parent is involved, and often the daughter cell resembles the mother cell. So we go to internal and external structures of seeds. Someone is putting up the hand and doesn't want to put it down. Please, are you putting up the hands by mistake? Because you forgot to lower them down, or you have something to say? If you don't have anything to say, or you just said it, lower down the hand. iPhone Pro Max, Jackie Kasana. Can you lower down the hands? Okay. Jackie? Okay, seems she forgot. So let's go to internal and external structures of the seeds. Here the question is identify the structural differences between the bean seed and the maize grain. So who the son said? Faith. Faith said she wanted to share something on this, so she could use this chance. Can I identify structural differences between the bean seed and the maize seed? By putting up the hand. Differences between the bean seed and the maize seed? Yes. Sharif?
Yes.
Who is going first?
Master, me what I know. A bean seed has two dicotyledon, has two cotyledon. Then
Yes.
a maize has one cotyledon.
Good. That's a good difference. We can note it. A bean leaf, a bean seed has two embryonic leaves, while the maize seed has one embryonic leaf. Who else? Please, after talking, you lower down the hand and you mute. Yes?
Okay.
Yep, Villa.
Yeah, another difference.
Yes?
Hello. Yeah, good afternoon, sir.
Good afternoon. Just straight I'll go to the difference.
Yeah. Another difference is the
Is it my network or yours? I'm not getting you well. Okay, Trevor.
Okay, for me, I can say that a bean seed has epigyl germination, then a maize seed has apogee germination.
Okay, what if we say that it undergoes instead of has?
Yes, be good.
But also another problem is here we're talking about structural, meaning structural means the way they are. Okay? So that one is not structural. That is not structural.
Okay.
Lower down the hand and mute. Yes, Aria?
A maize seed has an endosperm, whereby it's an endospermic seed, whereby a bean seed has no endosperm and it's a non-endospermic seed.
Okay, what's the role of endosperm?
It stores food for the seed. And
Okay,
for the
that's
seeds
good.
that
You want to continue talking?
yes, and for the seeds that have no endosperm, they store their food in the the cotyledon?
Okay, okay, that's a good one. Lower down the hand and mute. Melvin?
Sir, I think the bean seed has a hilum. Is it hilum? While maize does not.
Does not have. Okay, good observation according to you there. Any other? Please, when I unmute you, you talk immediately. Techno KL5.
A bean seed has one cotyledon while a maize seed. A bean seed has two cotyledons while a maize seed has one cotyledon.
Good, but we began with that. When they have mentioned it, you don't put up the hand again. Okay, let me pick last person here.
A maize seed has 2 scars and a bean seed has 1 scar.
Wow, good observation there. That's why maize seed is called a fruit, while a bean seed is not called a fruit because a maize seed has 2 scars. Okay, can we mute now? Okay, Bonita? Okay, so let's proceed. Here we have seen the external features, then the internal features. Okay, external features refer to those features which are found outside the seed. For example, tester. Tester is the outside covering of the bean seed, and then position of radical. Here it means that at this point, this is where the roots shall emerge. And then the micropyle is a hole which allows in air. And then hilum is that point of attachment to the pod. To the, we call it the placenta. And then the internal features of a bean seed, we have the plumule here. Plumule grows into the shoot system. And then the radical grows into the root system. The outside part is called the tester, and then you have the cotyledons. Yes, talk.
Isn't it the micropyle which also allows in water?
Actually, it also allows, though water can enter by osmosis all over, but also there it can enter. It's okay. Okay, so let's proceed to the internal and external structures of the of seeds. And here we are seeing a maize seed. Yes, Sharif?
Master,
Sharif?
someone talked about the scar. Someone talked about
Yes.
the scars.
Yes.
I never got it very well. Can you please elaborate
Okay,
more?
let me show
And
you
you
here.
said that the other is. Thank you, sir.
Listen, I show them here during this. Okay. So for maize seed, it has two scars. It has what you call the stalk scar and the stylo scar. So I have this one. The stalk scar is this one which attaches the maize seed, but in this case it is a fruit. We often call it a fruit, though here we are calling it a seed. So it is a fruit because it has this position of attachment to the cob, which you call the stalk scar, and then this scar up here we call it the stylo scar, which shows where the stylo pinched off from. Because the maize seed has these two scars, this stalk scar and then the stylo scar. That's why we refer to it as a fruit. Okay, while the bean seed is not called a fruit. So here we have the position of the embryo here, then position of the cotyledon here, and then testa fused with fruit wall. Then its internal structures. We have the position of the attachment to the cob, which you call the stalk scar. Then the stylo scar could be here. Then the whole of this part is the endosperm. And then this one, the plumule sheath, or sometimes called the chorioryza. Inside the chorioryza is the plumule. Then here we also have the chorioryza. Sorry, there was a mix-up here. This plumule sheath is called the chorioptile, not the chorioryza. We'll put that right. And then this radical sheath is called the chorioryza. So this is wrong. This is supposed to be the chorioptile. And then we have this the cotyledon, and then fused fruit wall and testa. Ravenna?
Teacher, on a fruit, I don't see any style scar. I see the stalk scar.
Do you have the maize seed there with you?
Teacher, not maize seed. As in, why is it called a fruit? Yet, teacher, what do you mean by a fruit? Like a mango fruit or what? That's why it's referred to as a fruit.
Okay, now, for something to qualify to be a fruit, okay, it should be having two scars. Okay? For example, when you are classifying something into a fruit, I'll give you an example. A mango. Okay? I hope you're picturing a mango in your, I know you've ever seen it. There is that scar which attaches to the stem, not so?
Yes.
But at the end of it, this lower side, there is also a scar which can be seen when it is still just emerging. The same to the banana. It can be ripe or even these normal ones. There is that point when it attaches to the stem.
Yes.
Okay. Then there is also another scar this side. You ever seen it?
Teacher, I'm picturing only that scar to the stem. The other one I don't know.
Okay, let me show you here. Let me show you. Okay, let's use a ripe banana. Can you see my screen with bananas?
Yes.
Can you see my screen with bananas?
Yes, teacher.
You see this side. You're talking about this side, right?
Yes.
This side where I'm pointing, not so.
Yes.
But there is also this side. Okay, this one can't be seen clearly. I hope it loads very fast. Now there is this side here.
Yes. Yes.
You see this scar?
Yes.
So this one is called the stalk scar, then this one is called the stylo scar.
Okay.
And because it has two scars, that's why we refer to it as a fruit.
Oh.
Actually, to simply call it, we call it a berry.
Mm.
Okay?
Yes.
So the same to the maize seed also. A maize seed has two scars. And it has the stalk scar and the stylo scar. That's why we call it a fruit also. So anything which has two scars, this side and this side, the stalk scar and the stylo scar, we refer to it as a fruit.
Teacher, on a mango, where is the style scar? There's only a stalk scar.
Of course, you only see it when it's still young. As it grows, the stalk scar goes, the stylo scar fades away. You can't easily see it.
Okay, sir.
Yeah, but for a fruit like, I think, an apple, the apple it is maintained. Is it? Yeah, for the apple, I think it is maintained.
Yes.
For most of them, they are maintained. Like which other fruit? But a number of them, they are maintained, I know. Even for the mango, even for the orange and the lemons, it can be seen, that point where the stylo scar was.
Yes.
So when something has two scars, then that is when we call it a fruit. Actually, the only way you answer, for example, if you're doing a practical and then they ask you that, where do we classify this? If it is a fruit, then the only reason you will say is it has two scars, the stylo scar and the stalk scar.
Okay, sir. Thank you.
All right. iPhone Pro Max. No, there is Aria.
Okay, sir. You've said that an apple is a fruit, but on what I recall, about what I studied, they say that an apple is a false fruit, so it's not a fruit.
Is that what?
A false fruit.
A false fruit.
Yes.
Okay, so when you're classifying fruits, ideally we classify them. But the first thing to classify something, to classify something as a fruit is, it should be having two scars. That's one thing we should always identify. If it is not having two scars, then it is not a fruit. Okay? Now, we further classify them into two also, into whether they are dry or fleshy. So an apple is a fresh fruit. However, it is not a drupe and neither is it a berry. But it lies in a group which we call a pome, P-O-M-E. I think we shall do that one when we were supposed to do that one in senior 2 when we were classifying under flowering plants. I'm sorry that we missed it. Maybe there is a Zoom lesson on that. I think I did it at the start with the senior tools. You can go to YouTube on Holistic Learning Platform, then you follow up on that lesson.
Actually, I learned about that. Yes.
All right.
Thank you, sir.
Josh?
Sir, is there any other way we can classify the fruits without saying about the scars?
No, the scale is the only way. It's just like saying that how can we classify that someone is a Ugandan without saying that they are Ugandan? It doesn't make sense, right?
Yes. Thank you, sir.
For something to qualify to be a fruit, it must have two scars. That's the first thing. Then we can use other reasons to classify them, but still under fruits. Okay?
Thank you, sir.
Yes. iPhone Pro Max. Aria, lower down the hand. I think you talked.
Excuse me, sir.
Yes.
Someone has talked about a false fruit. So how do I know that this is a false fruit and this is not a false fruit?
But we said the bare minimum of something being a fruit is 2 scars. Okay?
Yeah.
So
Yes.
it would be the maize, which we would call a false fruit, maize. But already it is because you can also call it a seed. Okay?
Yes.
So a maize, the maize seed is a fruit because it has 2 scars. So because it is also a seed, that's when you can call it a false fruit. But the apple is already a fruit because it has all characteristics.
Yes. Don't.
There is this one. Please
Teacher.
unmute and proceed very fast.
Teacher.
Yes.
Where do the scars come from?
The scars come. Have you ever seen a flower? Did you draw a structure of a flower? Oh, he went away. I wanted to ask you yourself, and then you understand best. But you see, when you see a structure of a flower, remember, fruit comes from the ovary. Now, on the ovary there is a tube which connects from the stigma up, and that tube is called the style. Okay. Now, when the flower after fertilization, there are events which take place after fertilization. One, the stylo withers off. Now, that point where the stylo withers off and falls off, it remains as a scar, which we call the stylo scar. While this lower part, where the ovary is attached, it is attached to something we call a peduncle, or we sometimes call it the flower stalk. So that flower stalk remains always as the stalk. And when you're picking the fruit, you often pick it from the stalk. And because that point, when you pluck off the fruit from its attachment, it shall leave the scar we call a stalk or scar. That's why often in most fruits, you find that the stylo scar disappears. Just like someone asked that, where is it in the mango fruit? Because remember, it is the first one which withers off. Therefore, after ripening, it can completely go away. While for the stalk, it usually, because you pluck it off from there, so usually it is still fresh, so you can easily see it. Okay, last one here. Samsung SM. Okay, they have gone to doze off. Let's proceed, I think. Samsung.
Hello, teacher.
Yes.
Teacher, I would like to know the difference between berries and groups.
Groups have one seed, while berries have many seeds. So, like when you see a banana, have you ever opened a banana in the middle, longitudinally? Like when you cut this banana from this side coming down here, you'll see some blacker things there, very many of them. Those are the seeds. That's why you call it a berry. Then a mango has one seed, therefore it is a drupe.
Okay, sir.
Someone called dash here. Please talk. I just see dashes on your name, so okay. So you can proceed now. Let's go to growth and development of the flowering plant. And in this case, in the flowering plant, development begins with the growth of a zygote into a simple embryo within a seed. So the embryo is differentiated into an embryonic shoot, which is known as the plumule, and the embryonic root, which is known as the radicle. So in endospermic seeds, such as the maize seeds, food for the embryo is stored in the endosperm, and here an example is maize. Then for non-endospermic seeds like the beans, here food is stored in the large fleshy cotyledons. So here we have these two words, hypocotyle and then epicotyle. Hypocotyle is the base of the radicle, and then epicotyle is the base of the plumule. Should you meet them? So let's go to seed germination. So seed germination is a process by which the embryo of a seed grows into a seedling capable of existing as a new and independent plant under favorable conditions. So we have here seed coat, plumule. We have hypocotyle there. We have the hilum. We have the micropyle, and then we have the cotyledon. I've already seen this. So let's go through types of germination. Maybe can someone try to relate for us epigyl germination, the way they have ever learned about it before? I'm sure this is not the first time we are meeting it. Okay, Faith is ready to take us through. Faith shall take us through epigyl, then Einstein. I saw Einstein. Okay. A boy shall take us through hypogyl. No, first wait. Faith. A girl shall take us through epigyl, and then a boy shall take us through hypogyl, and then another person randomly shall tell us the differences. So, Faith, you're starting with epigyl.
Good afternoon, teacher.
Good afternoon to you.
What I know about epigynation? This is a process where the cotyledon appears on the ground, and it's due to the rapid elongation of the hypocotyle.
Yes.
Yeah, that's what I know about it.
Yeah, actually, I've talked that it occurs here and like that.
No,
okay.
I know it's a it's where the cotyledon appears on top of the ground due to the rapid elongation of the hypocotyle.
Okay. Where in which cities does it take place?
It happens in what dicotyledonous seeds.
Like?
Beans.
Okay.
Yeah.
Okay. Another girl to try to. Okay, we thank her. She has done a great role there. Is there someone who thinks they have something to complement or supplement on what Faith has just said? Definitely a girl also. Is Riven a girl?
Yes, I'm a girl.
Uh-huh. Do you have something to add on what she said?
She had same knowledge that the cotyledon appears above the ground. That's what I know.
Okay, I want someone who has something to add on hers. Aria, I hope you're a girl also. You have none? Okay, let's go to Jovan to take us through hypogyl germination. Jovan?
Yes, teacher. Thanks for the opportunity. Teacher, hypogyl germination, this is the process of germination where the cotyledon remains below the ground.
Yes.
And this type of germination takes place in monocots, such as the maize seed, the sorghum, and it's due to the elongation of the epicotyle.
Okay, great. Do you have something to add?
I think that's it.
Okay, thank you for your contribution. No, I remember Melvin being a boy. So Melvin, add something on what he said.
So, discussing about the process of hypogyl germination.
Yes.
So, what hypogyl is that the hypogyl germination, the Actually, made happens in monocotyledonous seeds.
Okay.
And during this type of germination, the seed undergoes imbibition in order to soften the seed coat.
Yes.
The the plumule, the first piece, comes above the ground, leaving
Yes.
the and the endos cotyledon, something like that. It leaves its way out of the ground. Yeah.
That is it, eh? Okay, thank you for that addition. Has brought us a new word called imbibition. Someone shall tell us the meaning of that. Let me check someone who has not talked today. Gemma? Is it Gemma or Jemima?
It's both, but I just cut my name short.
Yes?
I just cut my name short, sir. So I
You
don't
did
know
what?
the meaning. I don't know the meaning, but I had a question.
Okay.
So our master told us that epigeal germination is the type of germination where the cotyledon appears above the ground due to rapid elongation of the hypocotyle and epicotyle. But the first speaker mentioned only the hypocotyle. So is it both hypocotyle and epicotyle or one?
No, hypocotyle elongates faster than the epicotyle.
But should both of them be listed in an examination?
Should both of them be
listed?
Be mentioned
in the definition.
Now, okay, I know you're adopting from the old curriculum where you're still having this thing of definitions in the mind. Like in new curriculum, we don't have definitions, but we have meanings of words. Okay? In other words, you should understand the meaning of a word, not simply cramming the definition. You get it?
Yes.
Yeah. So when you start in the definition, it doesn't sound okay. But if you know the meaning that the hypocotyle elongates faster in the epigeal germination, then the epicotyle elongates faster in the hypogel germination, then it's better.
Oh, thank you, sir.
All right. There is checking on ones who have not talked yet. So if you've been talking, I'm sorry. Right on 23. Unmute.
Good afternoon, sir.
Good afternoon to you.
Okay, thank you for the opportunity. I would like to contribute on germination in the maize.
Yes.
Yeah. The process undergoes 6 stages where we have the imbibition, activation, radical emergency, plumule emergency, cotyledon expansion, and seedling establishment. You asked a question about imbibition.
Yes.
Imbibition, this is where the water enters the seed through micropyle.
Yes.
And this softens the seed coat.
Yes.
So from there we go to activation where starches are broken down into sugars for energy. That is activation.
Yes.
And then the radicals, they grow downward into the soil and the radical emergency.
Yes.
Then primary emergence, this is where the shoot system grows upwards towards the light. Then here in the cotyledon expansion, the leaves, small leaves starts emerging as they unfold. Then finally, the roots deepen and leaves expand, and then the coat shades here under seedling establishment.
Thank you. You took us through the whole process, which is very good. Imbibition is the taking in of water. Activation, the water activates the enzymes, which break down food. Who has not muted there?
Shanita.
Mute. Okay, so Rayton took us through the whole process, which is good for us to know. Though those processes take place generally in all of them, both the dicot and the monocot. Maybe we shall try to break them down. Rima?
Yes, good afternoon, sir.
Good afternoon to you.
Yeah, I would like to take us through the difference between hypogea and the hypogea germination.
Yes, do.
The first difference is that in the hypogea germination, we have in hypogea germination, the cotyledons remain below the surface of land or earth. And then the second difference is that the hypocotyle of in epigel germination, the hypocotyle elongates, while in hypogel germination we have epicotyle elongates. And then under epigel germination we have actually, yes, I think those are the one which I can try to give the difference.
Okay, can someone comment on his work and definitely improve it? Raivena or Rivena, I don't know.
Teacher, for me, I have a question and I'm very confused about it. In epigel germination, you said the hypocotyle elongates faster than the epicotyle. And I thought, and the hypocotyle is at the base of the radicle. So I thought the epicotyle is the one that elongates first to enable the cotyledon to be seen above the ground, since it's at the plumule, which is at the top. But you're saying the one at the radicle is one that elongates. Is it to push the cotyledon up over?
Okay, I hope this has drawings. Let me check if it has drawings. This one shall help us explain. I will answer that when we reach here. Okay?
Yes.
Josephine?
Good afternoon, teacher.
Good afternoon.
I would like to add on the differences of epigel and hypogel germination.
Please go ahead.
The first point is in epigel germination, the epicotyle is short in this germination, while in the hypogermination, the epicotyle shows rapid elongation. The second point is in epigel germination, cotyledons turn green and carry out photosynthesis, while in hypogel germination, cotyledons don't undergo photosynthesis. The last point is in epigel germination, the upper part of the hypocotyle, is curved and to protect the delicate plumule, while in the hypogel germination, the epicotyle shows no curvature.
Okay.
That's all. Thank you.
Okay, thank you. Thank you for those wonderful views concerning germination. So let's go through it. You can now lower down the hands. So here, this one clearly shows us the hypocotyle. radical axis here, which is being shown here. Then this is the plumule. So during growth, this hypocotyl elongates. When it elongates, it forms some sort of a curve. Okay. So that's why someone was confusing it up. So this is what they were meaning. This is the hypocotyl. Then the hypocotyl here is elongating. Okay. But then when it elongates, you see it tries to push through the soil, therefore helping it come out. Then when it comes out, then it will definitely pull the cotyledons out. Therefore, them opening to give the embryonic leaves. So this is where the cotyledon appear above the ground due to rapid elongation of the hypocotyl. This one takes place in beans, tomatoes, cotton, mangoes. So cotyledon is mainly being small. Once exposure to sunlight, turn green and become photosynthetic. This is because they contain chlorophyll. This is when they assume the function of making food. So in this type of germination, the upper part of the hypocotyl is hooked, like you see this hook here. It is hooked and is all curved downwards as it grows through the soil to protect the delicate plumule. So I hope someone was asking has got that clearly. Then under hypogyle, here the epicotyl grows faster than the hypocotyls. And then seeds showing hypogyle germination are called endospermic seeds, like maize seeds and nuts. The seeds germinating this way often have large cotyledons to nourish the growing embryo. Like you see, they are always, remember for maize, they always have the endosperm. So, but they also have the large cotyledons to nourish the embryo until the first green leaves appear so that they can photosynthesize and make their own food. So let's go through the differences here. Under epigyle, cotyledons emerge out of the soil, and at the other side they do not, as they remain inside. Hypocotyls also show rapid elongation, while in hypogyle they don't.
Excuse me, teacher.
Yes.
I'm kindly requesting you to help me, go back to the previous page and I just take a screenshot.
I'm going to share these notes, so I want you to only listen and understand. Don't worry about it. Or you don't have data to download them.
I have.
Then you'll get them at the end of the lesson. I'm going to share the PDF in the chat room.
Thank you, teacher.
Blessing?
Teacher, I was asking,
Yes.
why does the hypocotyl turn like it's green when it's still under like the soil? It's not yet exposed to the sun. How is that possible?
Come again?
Come again? I was asking, like in this picture, it shows that the hypocotyl is already like becoming green and it's still under the soil. It's not yet exposed to sunlight. Yes.
And so you're asking what is wrong with that? Okay, I hope that one is not an issue. Where is it turning? Okay. Remember, hypocotyl is just more of a protective layer for the plumule. So it comes out. When it comes out, then it shall give chance for the cotyledon to also come out. Of course, you see it turning green here because when it comes out, it shall be used for photosynthesis. But I don't think this one would be an issue, though. Okay, hey.
Well, thank you.
Teacher, why do they mainly compare a bean and a maize? Can they also compare other dicots and monocots?
Of course, you know that these are the most common, okay? I'm sure all of you here have ever seen maize and beans. So who has never seen them?
Yeah. Teacher, you have also seen rice and cowpeas.
Not all places have rice.
But everyone has eaten
But when
rice.
it's okay, for you can compare others, it is okay. It's okay.
And also, teacher.
Yes. I can hear you. Yes?
Monocots are fruits.
Come again?
Are all monocots fruits? Are all monocots fruits?
No.
Are
I know of the maize. Okay. Like, of course, you get usually many differences. For example, like there are many usually, like we usually generalize them, but there are some differences which happen down there as we study them further. But for now, of course, the only fruit we know is the maize, which is a false fruit, but we can also call it, actually, it is commonly called a seed. So rather, it is a false seed than a fruit.
Teacher, don't the other monocots have scars, like have two scars?
Maybe you can help search that out and you tell us.
Okay, sir.
Yeah.
Thank you, teacher.
All right. iPhone 17.
So I was saying that you helped me on hypogermination, the reason why the cotyledons remain in the ground.
Of course, the reason as why the cotyledons are coming out of the ground in the
No, on hypogea, why they remain in the ground.
Of course, I'm starting with epigel. In epigel, the reason as why they remain, they go up is because of the hypocotyl elongating. Okay? But it is the opposite in the hypogel, where the epicotyl instead elongates. Because it is elongating, therefore it means that there is no way it can come out. So here the reason as why in the epigel it is coming out is because of the elongation of the hypocotyl. While in epigel it is the epicotyl which is elongating, like you see here. Okay?
Oh, okay.
That's why it remains downwards. iPhone 17, Samsung. So the Samsung that I talked should first lower down the hand so that I understand which one was not talked.
Hello, teacher.
Yes.
I'd like to know the conditions that facilitate the elongation of hypocotyl in epigel germination.
The reason as why it elongates is there are hormones, call them plant hormones. In their presence, they cause elongation or they cause growth by causing division of cells, which increases the size. So the growth is simply coming out because of the hormones, which you call auxin.
Okay, sir.
Okay, we can proceed now. So these were the differences. I'm going to share the notes so you can pick them up there. Then let's go to physiology of germination, which someone told us earlier. So the first point is called imbibition, like someone told us. Imbibition here, the seed takes in water through, it can be through the whole tester, like I say, or it can be through the micropyle. And then, so that is done, that process of the seed taking in what is called imbibition. And then The second step is called activation. The presence of water activates enzymes. As enzymes usually be there, but in their inactive state. So the presence of water makes them active. And when they are active, it means that they will help in the next stage, where they will break down the stored food. Here in the maize, it is stored as endosperm. So the enzymes, for example, here enzyme diastase, shall catalyze the conversion of starch to glucose. Proteins are also converted to amino acids by protease, and then lipids to fatty acids by lipase. So the first stage was taking in what we called it imbibition. Second stage is activation. Third stage is breaking down the starch, proteins, and fats which were stored in the seed into their simpler forms. Starch being broken down into glucose, proteins being broken down into amino acids, lipids being broken down into fatty acids and glycerol. And then water dissolves the food nutrients and transports them into the growth center, the embryo. And then glucose, fatty acids, and glycerol are respired to provide the energy, which is responsible for cell division, while amino acids are used for growth of the embryo. And then the softened tester ruptures. Of course, it tears away, and therefore allowing the radical to emerge first, followed by the plumule. Someone whose hand is up, I hope you're not from the previous. Samsung?
Okay, teacher.
Yes.
I'd like to know when someone talked about inhibition.
Yes.
He also talked about activation.
Yes.
How does it take place?
How do they take place, the two?
Yes, sir.
We said imbibition is the time when the seed takes in water, either through the micropyle or through the tester. Then the presence of the water activates enzymes, because at first when the seed is still in the store, the enzymes are there, but they are in their inactive state. State. Therefore, when you take in water, you activate the enzymes into their active state.
Thank you, sir.
Toyota? Toyota?
So I would like to take us through the conditions necessary for germination.
You want to take her through?
Yes.
Okay, do please.
Okay. Some of the conditions necessary for germination. There is air, which contains oxygen and is necessary for the production of energy needed for the germination through respiration. Then water activates the enzymes within the seed to catalyze hydrolysis of the stored food into simple compounds. And temperature, which is and temperature. Though I'm not sure what temperature is how the temperature works, like what it's required for.
Okay.
Thank you.
So okay, good idea there. She has talked some good points there concerning the conditions. Eli Ari?
Teacher, I want to add on one condition that she has left out. The condition is oxygen.
She began with that. Maybe just said I want to bring it in another dimension.
Okay, teacher.
No, you talk about it. Okay. Irene? Irene?
Yes, hello, Master.
Yes.
So I was asking, is it possible for you to send these notes to us?
I'm going to send them here on this Zoom. Actually, let me send them now. We are still continuing,
Thank you,
by the
Master.
way. We are still continuing. Let me share them. As Sharif talks, I wish you're talking about the conditions.
No, Master.
What do you want to talk about?
Just I was requesting before the lesson ends that Master, as we are going back to school, that would you please get us some scenarios concerning about this work that you have taught us?
Okay.
Like revision.
I will share some scenarios in a group.
Yes, sir.
For past papers.
But it will be good also if you take us through.
Through what? Through them, eh? Definitely, of course, with the extent of the holiday, I hope we meet next week also. We can meet it more so for candidate classes for the interface on how to attempt the questions. I think that one would be better for us.
Yes.
Okay? Yes. Okay, the notes are coming along, so. Okay, there they are. They are coming up. They are coming your way. Oh, all this want to talk, iPhone, Solomon, Samsung, Rayton. Yes?
Master.
Yes.
Me, I'll be glad if you bring in that point you have talked about.
Come again?
That point of question approach. I'll be glad if you bring that point of
Yeah.
question approach next week.
Yeah,
Because it is
we shall
better.
do that next time. Should we have another session? We shall just be meeting for approach on how to attempt those scenarios.
Yes, sir, because that is the challenge we are facing.
Yeah, definitely. Shall do that. So I've also shared the notes here on the group chat. iPhone 17.
Master, they are talking about the conditions necessary for seed germination.
Yes.
But I've heard of that there are some external environmental conditions and internal seed conditions.
Yes.
Now, I'd like to know the internal seed conditions.
Come again?
I would like to know the internal seed conditions and explain them to me.
Now, in that case, of course, now the internal usually are the. I will usually relate that when the seed fails to germinate, maybe. For example, when the embryo is still immature, maybe the enzymes fail to get activated. But usually here we talk about the general ones like we see here. Okay? Most of them are general. For example, warmth. Warmth is important for, we know that enzymes work within a narrow temperature range, above which they are denatured and below which they are deactivated. So that's why warmth is very important. For example, oxygen is important because there must be the glucose which is converted, which is converted from starch, and then the fatty acids must be used in the process of making energy through respiration. And you know that aerobic respiration only takes place in the presence of oxygen. That's why oxygen is important. Water is important because one, it's smooth. It makes the tester weak. Therefore, the hypocortical can easily break through. But also, secondly, it also helps in the activation of enzymes, such as they can break down stored food into the usable forms. Okay.
Okay, Master. Yes.
You can now mute and lower down the hand. Jovan?
Yes, teacher. Teacher, I would like to ask you if we shall meet next week since the holiday was extended.
Yeah, that's what I was contemplating, but you will get notified should it be there next week, and then we shall simply handle question approach.
Okay, teacher, we shall
Or
be very
item
glad.
approach. Yeah. Samsung?
Excuse me, sir. When our teacher was teaching us, he told us on the conditions necessary for germination. He also mentioned about light, because there are some seeds which require light for such germination like that. Oh, I don't remember well, but I remember like
Yeah,
the
that is very
photoblastic
true, eh?
seeds.
That is very true for photoblastic seeds. There are seeds which, also small seeds, like seeds of eucalyptus. There is some amount of length of exposure to light they require before they can germinate. If it is below or above, then they will not.
Okay, so if I'm
So
writing
that is also
conditions,
true.
excuse me, if I'm writing the conditions, for example, and they have asked me, can I write that light?
The first major one was at D3. The other one is just for photoblastic seeds. And I'm imagining, should it be a practical? There is no way they will bring a photoblastic seed because it shall be way too small to be seen.
Oh, okay.
That's why we consider these three. Yeah.
Okay.
Right on.
Thank you, sir. You had forgotten about the soil.
Oh, soil as a factor, as a condition.
Yes. Without
But have you ever put some seeds in your house, cemented, and then you pour there and then you see them germinating?
they have, but what
Is
they taught us
soil
is
there?
they taught us that that soil also is a factor whereby the proper pH in the soil also ensures the nutrient availability, thus enabling the seeds to germinate. And also soil also support the seedlings.
Yeah, that is very true, eh?
Yeah,
That
and
is
another
very
thing
true,
that
but those ones are secondary, eh? In other words, should you put the seeds in the soil without water, then they will not germinate. Okay? So that's why those ones just become secondary. But the first primary ones which must be available before germination takes place are these ones.
Okay, master.
Oh, you are continuing. Sorry, I muted you. Continue.
I don't have the timetable for the lessons, so I kindly request you to drop it to me.
Okay, my co-host shall share it. I hope my co-host shall help share it. But are you in a group? Are you in a group?
Yes.
Then you can access them from there if you're in the group. Okay, iPhone 17 Pro Max. Be quick. iPhone, okay. She's not there. Seguya, but these are people who have already talked, I think. Solomon.
Excuse me, teacher.
Yes.
Teacher, a certain speaker spoke about the requirement of light for photoblastic seeds, yeah?
Yes.
Teacher, now what's mainly the importance of that light? Or what
Actually,
role
the
does
importance
light
of the light, actually, the thing is not light, but the length of exposure to light, actually. It is supposed to be like that.
Yes.
The factor is the length of exposure to light by the seeds, not simply light. Whereby if those particular
Yes.
seeds require some length of exposure to light before they can germinate, and should that length not be reached, then they'll not germinate. But that is minor. It occurs only in some small seeds, not all of them. That's why it is brought as a last factor, not you shouldn't begin with it.
Yes, teacher. Thank.
Excuse me,
Okay, I
teacher.
want our host to tell us something. Yes.
Teacher, someone in the group was asking you to reshare the notes. Sorry, in the chat. And then
Yeah, by the way, you'll help me go through those chat, because I don't pick up on the chat. Maybe should you find there any questions, you can help me bring them up.
okay. Then someone was asking you to explain more on germination. I think it can be in the next lesson.
My germination was so wide, so anyway, maybe they could have been specific. Okay, next one.
So that's it.
I remember the first question you began with. I don't know.
Someone was asking you to reshare the notes.
Oh.
Yeah.
But also, do you have the timetable? Maybe you help share with them.
I think I can get it.
Okay, then you can help me share it here. Okay, there they come. Please download them before we go off. Otherwise, when you go off, you'll not be able to see them. Please be quick when you have a question.
Teacher, what if all these
Yes.
conditions, they are around and the seed fails to germinate?
Now, that's when you bring in internal factors. For example, the embryo is immature. Maybe like there are some chemicals which are present in the seed. For example, abscisic acid is too much. Okay.
Yes.
So those ones can also, or the seed is literally also not in good condition. Maybe it has holes. It has the embryo has been denatured like that by pests.
Oh, teacher, is that related to this thing of seed dormants?
Exactly.
Oh, and also, teacher, is there this thing like it has insufficient nutrients?
Sufficient. Now that when we talk about in terms of chemical substances present in the seed, because for a seed to germinate, it must be having gibberellic acid. Should the gibberellic acid not be enough, then the germination won't take place.
Oh, thank you, teacher.
All right.
Sir.
Yes.
Sir.
Yes.
I am just asking on the platform. We are not seeing the work on the platform, and they are saying
this
that
work I'm covering.
Yes.
No, this work is got in the chat room. This chat, you check the chat room for this Zoom, then you download it.
Okay. Thank
you. Ellen, is it Eli Ari? iPhone 17, Solomon, Are you there to talk? Or you just put up the hands and you forgot? Okay, I hope today we have met. I wish we meet again, but should we not, then I will wish you the best until we meet again, hopefully. We pray God keeps us well. Meanwhile, let's revise. It's only through revising, and not just, some people just read, they don't know how to revise. They revise because other people are revising. They revise because they are being forced by parents to revise. Others, they revise to pretend, say that they can pretend in that and they don't do house chores. That is wrong, eh? Revising is, you intend it by yourself, like it is intentional, where you must pay attention to what you're revising and then you make summaries of them. By doing that, you'll help improve your grades. While if you're revising to make people happy, you'll not improve the grades. So be serious, use the holiday and all the free time you have to revise to improve the grades. I hope I can see you next year in A level, and then in very few years you become doctors, others medics, others can also become teachers. Only for whichever career you want to take after this. Otherwise, I wish you the best. We know we shall meet soon. Otherwise, I only wish you to be serious and then you revise. And this part, they still have something to say. I see the hands are still up. Irene, Ari, Solomon.
Excuse me, teacher.
Yes.
My concern is in this part of temperature. That what if the temperature is below 10 degrees Celsius and above 50 degrees Celsius?
Below 10 degrees Celsius, the enzymes shall become inactive or they will not be able to work. Above 50 degrees Celsius, the enzymes shall be denatured. In others, they will not be able to work again. They will be destroyed.
Okay, teacher.
Please unmute very fast. Should you be having something to say before I go?
Sir,
Yes.
is it true that, like, as in the chapter of seed dormants has been removed from our curriculum?
Understand what you must understand, okay? All this is curated using the NCDC book, eh? So what is not there, then it is not part of the curriculum. Any other person? Yes.
Me. Can we get other subjects apart from these weekly subjects that we are getting?
Like which subjects haven't you met so far?
Like history, like
we are going to
geography.
have all of them with time. Okay?
Yes.
Definitely, we pray one time. We can even have a full class here from morning to evening. Hopefully, one time.
Okay, let's pray.
Yeah. Okay, I think that's the end. I wish you the best. Have a good evening. Keep safe. See you soon. Bye.
Chair prayers.
Oh, you can pray for us. It's okay.
Okay, then thank you so much, teacher. Members, let's humble ourselves and we pray. Loving Father, King of Glory, we thank you so much for today. Thanks so much for the lesson that you have had today. Thank you so much for the teacher and everyone who have participated today. We pray that may next time also be the same. We thank you for the good network. May it always be the same. In Jesus' mighty name I pray and believe. Amen.
Okay, perfect. Amen. See you. Bye.